Thank you, and thank you for having me here. One of the fun things about starting a job at Emory is that it is in Atlanta, which has this really rich scientific community at multiple colleges. So it's fun to get to be here, so thank you again for having me. So I am going to tell you about evolutionary conservation of a social subcircuit in the brain that promotes sociality. So we see an extraordinary amount of diversity in social behavior. Certainly across species, you've got some species that are, sorry, try not to turn too far away from the mic. We've got some species that are incredibly social. They live in large groups, often in groups of hundreds or more. And then you can kind of think of sociality as a continuum, such that while you've got some of those really social species, you can make your way down and have some moderately social species all the way to the other end where you've got animals that are extremely territorial or antisocial. But even within a species, there's often a great deal of individual variation in social behavior phenotype. So I'm interested in the mechanisms that underlie social behavior and also about how that variation in social phenotype arises. So one of the big questions that my research addresses is how did the brain evolve to produce variation in social phenotype, such that we can get two species of closely related birds like the zebra finch and the violated waxbill. So both of these species are strilded finches. They both exhibit social monogamy, so they both are forming pair bonds with one mate. They're also similar when it comes to their parental care, such that they're both biparental. Both mothers and fathers take care of the young. But how they differ in their social structure is their grouping preferences. So the zebra finches, extremely social and gregarious, whereas those violated waxbills are extremely territorial, and they don't really like anybody except maybe sometimes their pair bond partner. So if we can understand what in the brain underlies prosocial behavior in both of these species with different phenotypes, then we might be able to get an idea of some mechanisms that may be fundamental to promoting prosocial behavior across species. Sorry, it kind of sounds like I'm cutting in and out. Would it be easier if I... One moment, please. Try not to get my hair out of the way. How about that? Better? Maybe. Good enough? I'll only turn this way. Okay. So, sorry, I'm pushing things around here. Another question is, do relevant neural mechanisms evolve in predictable ways when phenotypic convergence is seen in behavior? So here we're looking at, again, at two species of strilded finches, Angolan blue waxbills and zebra finches, and they have both independently evolved a more gregarious phenotype. So the question here is, have the same neural mechanisms evolved to promote this similar gregarious phenotype, or have they achieved a similar behavioral endpoint via different modifications to the brain? So to answer these questions, I use a comparative approach with a goal of identifying a mechanistic baseline of social behavior that may be present in all species. So throughout my talk, we'll see that there are some strongly conserved features of the social brain. And so we'll see similar mechanisms that promote social behavior across several different species. However, every animal and every species is going to face unique pressures that they have to respond to, whether they're environmental or social. And not surprisingly, animals typically respond to these challenges in different ways, which can push them away from that central tendency and result in deviations from that mean. So what's important here then is to add as much biodiversity as we can to our studies. The more species we study, the more likely we're going to be able to identify a mechanistic baseline that may be fundamental for social behavior in all species. So I'm going to talk a lot about social behavior, as you might guess. Social behavior is an umbrella term. It can mean a lot of different things. Technically, aggression is a type of social behavior. So there's one social behavior in particular that I'm going to be talking a lot about, which is sociality. So to give you an operational definition of how I'm using that, sociality is a general tendency to affiliate that is not directly related to reproduction. So I see sociality as a continuum such that we're going to have some species that are more social than others, and even within a species, some individuals that are more social, and even within an individual. There are certain contexts when an animal is only going to be so motivated to interact with another animal when they're not driven by the motivation to mate. So we'll see that there are some similar mechanisms that underlie reproductive social behavior as well as non-reproductive sociality. And, sorry, I'm distracting myself with the cutting in and out. And so while we see that there are some overlapping mechanisms, I still think it's really important to disentangle social context and to consider non-reproductive sociality as something that is distinct from reproductive behavior in a social context. Okay, if we want to study social behavior in the brain, what can we look at? The nonapeptides. So many of you are likely familiar with the mammalian nonapeptides, vasopressin and oxytocin. So across vertebrates, except for jawless vertebrates, all vertebrates are expressing some form of vasopressin and oxytocin. Now, while the nomenclature is taxon-specific, within a clade, each of these peptides differ by only a single amino acid. And because I'm going to be jumping around between different species across taxa, to keep things simple, I'm going to refer to all of the oxytocin forms as just oxytocin, and both of those vasopressin forms as just vasopressin. So for about 30 years, people have been studying the involvement of the nonapeptides in behavior, and we know that they are involved in a lot of different things, from affiliation and sociality, aggression, dominance, parental behaviors. They distinguish mating systems and voles, involved in stress response, as well as different types of communication and learning. So the nonapeptides are doing a lot of different things. Now, while the nonapeptides can be found throughout the brain, importantly for my studies, they're found in every region of something called the social behavior network. So across vertebrates, there is a network of interconnected structures throughout the basal forebrain and midbrain that are fundamental for numerous social behaviors. So here in this colorful diagram, each of these different colored circles represents a different region of the brain that's in this social behavior network. And you can see that they all map on and can be found throughout our vertebrate taxa. So you don't need to worry about what all these acronyms mean. We are going to focus in on two of these in particular. So a lot of my work has involved going into the brain and manipulating specific vasopressin and oxytocin cell groups within these different brain regions. And today we're going to take a look at just two of them, which we're going to see make up a prosocial subcircuit. Since we'll be looking at both birds and rodents, here we're looking at sagittal views of a bird and rodent brain. The first brain region of interest is a mouthful. It is the medial bag nucleus of the stria terminalis, which I will just refer to as the BSTM, shown here in purple. So the BSTM is a part of the medial extended amygdala. And then we're also going to be looking at the lateral septum, which is shown here in red. So just bird brains now. We've got a coronal section here. We're about midway through the brain. And our BSTM, shown in purple, contains vasopressin neurons that send axonal projections throughout the brain, but primarily to the lateral septum, which contains vasopressin and oxytocin receptors for that vasopressin to bind to. So I've been studying this subcircuit for a bit more than 10 years now. So first I want to tell you about some early studies that we've done that look at the anatomy and function of this circuit. So the BSTM-LS vasopressin circuit distinguishes social phenotype in estrildid finches. So again, we have 4 species of closely related estrildid finches that range selectively in their grouping behavior. So again, all these birds are biparental, they're all socially monogamous, but some of them are extremely gregarious and highly social, like the zebra finch, and they become progressively less social as we make our way down to that territorial violated waxbill. Now, if you take a look in the brain, and look at the number of vasopressin neurons in the BSTM and densities of those nonapeptide receptors, we see that our more social species have more vasopressin neurons in the BSTM and more nonapeptide receptors in the lateral septum, suggesting that these neurons may be involved in promoting that highly gregarious phenotype. We see more of them in the more social species and fewer of them in the less social species. Another study of anatomy is looking at courtship behavior, so reproductive social behavior in zebra finches. So zebra finch males, much like human males, some of them are excellent at courting individuals, and others are not so great at it. So we refer to these as duds and studs. So the males that are really good at courting females, our studs, have significantly more neurons in this cell group. So here we're just looking at the number of vasopressin neurons in the BSTM. The courters that are really good, they have a lot of these neurons compared to the males, the duds that don't have many. Suggesting that, okay, so this cell group may be involved in promoting not only gregariousness, but also courtship behavior. So if we then take a look at the other end of that circuit, the lateral septum, if you infuse vasopressin into the lateral septum, you see a decrease in aggression. And then if you were to go in and block vasopressin receptors in the lateral septum, then you see a decrease in gregariousness. So this suggests that that lateral septum also has properties that are promoting prosocial behavior while also decreasing aggression. Now, this is suggestive that that binding may be coming from vasopressin originating in the BSTM, but it's not a guarantee because the lateral septum can receive vasopressin from a lot of different cell groups in the brain. So from studies like this, we don't know yet for certain that that vasopressin binding is due to the BSTM vasopressin neurons. So a lot of the earlier work that I did based on limitations in technology, and especially technology in birds, because the viruses just don't work so well if you want to do viral vector manipulations in birds, it only allowed us to look at one part of the circuit at a time. And so that's something that I'll touch on later in the talk. Okay, so now we've taken a look at some anatomical findings. What about function of that BSTM vasopressin cell group? So we can ask about functional questions using something called immediate early genes. And so we can use these immediate early genes to examine neural activity. And so the IEG that we use in particular is called FOS, and it serves as an indirect marker of neural activity. So if we take a look at our photomicrograph here, we have a vasopressin neuron labeled in green and that FOS protein in red. And so we can see that our vasopressin neuron is expressing FOS in its nucleus, meaning that this neuron has recently responded to something. So what you can do is, you could take me, I could stand in this room all by myself, nice and quiet, think about life, and then have everybody suddenly appear. And if you look at my brain 90 minutes later, neurons that are showing FOS in the nucleus are neurons that have presumably responded to your sudden appearance. So this is something that you can easily take into the lab, not all of you guys to expose to birds, but for other conditions to get an idea of what these neurons are responding to. So with studies like this, we've shown that our BSTM vasopressin cell group is responding specifically to positive social stimuli. So what we did was we took male and female zebra finches, we put them in a control cage, or just an empty cage, or we exposed them to a novel same-sex conspecific. Sorry, keep cutting in and out here. On our y-axis here, we have the percentage of vasopressin neurons in the BSTM that were expressing FOS. So these are the neurons that are active. And you can see that both for males and females, these neurons are lighting up more when they're exposed to a social stimuli. But how do we know that it's positive social stimuli? I mean, they really like social interactions. So how do we know that that's different from, positive non-social stimuli? So we took it a step further, and this time we exposed our birds to momentary pause in time. Okay, never mind. Sure. So we exposed our birds either to that control empty cage, to a water bath, which they love. And so we had deprived them of open water to bathe in. So as soon as they had been deprived for a while, and then you give them a water bath, I mean, they literally just like dive in and start splashing around. They really like water baths. Or we expose them to that female. And you can see that these neurons are, again, they're significantly lighting up more, specifically to the positive social context. So they're not just lighting up to, positive stimuli. They're specifically lighting up to positive social stimuli, suggesting that there's something unique about this cell group for processing social components of behavior. The cell group responding to positive social stimuli. to really kind of hammer this point home, we can take that related species, the African violet-eared waxbill, which is highly territorial. So because they're territorial, we can presume that a same-sex conspecific, so if you expose a male to another male, that is presumably going to be a negative social stimuli. They immediately fight, they don't like each other. However, if you expose them to their pair bond partner, that would presumably be a positive social stimulus. And so with the violet-eared waxbills, expose them to the empty cage, to a male or to a female, and we see that this cell group actually decreases activity in response to that negative social stimulus, but increases activity in response to the positive social stimulus. So pretty cool that we can see some conserved functions across species here. However, what I've shown you about this subcircuit so far has been indirect or correlational. And I wanted to really hone in on the direct functions of this cell group, and so we needed to establish causation. So I wanted to know what are the direct contributions of the BSTM vasopressin cell group to behavior. So to do this, we used, nope, not yet. So based on those previous studies, I hypothesized that the vasopressin circuitry of the BSTM-LS is promoting male-female affiliation while also suppressing aggression. And I hypothesized this because they need to be able to behave appropriately in a reproductive context. It's not going to be in the best interest of a male to go court a female and immediately start beating her up. So to have a circuit in the brain that's going to be at the same time suppressing that aggression to allow and produce more prosocial behavior could make a lot of sense. Okay, so to test this, we needed to directly manipulate the BSTM vasopressin cell group, which we did using vasopressin antisense oligonucleotides. So antisense allows us to block translation of mRNA into actual peptide, allowing us to site-specifically knock down production of vasopressin. So in our picture here of a little zebrafinch, that cap sticking out of its head is a cannula, which if we return to our atlas here of a bird brain, this black circle on top represents that cannula, and then the two black lines dropping down into the brain are the two stainless steel cannula prongs that can be situated above your target of interest. So for us, we had them drop just above the BSTM, and then every 12 hours, you can go in and infuse this antisense, and it's going to continually knock down production of vasopressin so that there's just fewer vasopressin neurons there releasing peptide. Now the control for this is a scrambled oligo, which is literally just scrambling the gene sequence so that it doesn't bind to anything. So we take males and females. They receive either the vasopressin antisense or a control. And then we put them in colony observations. And so in a colony, we've got males and females. They get little nest cups, nesting material, food and water. And they get to hang out and kind of establish their own little birdie society over the course of a week. And then we go in and we do spot sampling morning and evening of every day so that we can obtain measurements of things like aggression and courtship, pair bonding behavior, general maintenance behaviors like grooming and feeding, as well as nesting behaviors. So from these colony observations, first we'll take a look at aggression. On our y-axis here, we have the number of aggressive behaviors directed towards same-sex individuals. And in green, we can see that our males that received the antisense and had this cell group knocked down were substantially and significantly more aggressive than males that received the control or females that received either the control or the antisense, suggesting that at least in male zebrafinches, that natural endogenous peptide in this brain region is indeed important for suppressing aggression, such that when you knock it down, they become more aggressive. Similarly, when we take a look at courtship behavior, so here we've got the number of male-directed songs to females. And so when they court females, I don't know if anybody's ever watched a zebrafinch, they kind of like dance back and forth on the perch and sing their little song. And so the males that had this cell group knocked down sung significantly fewer of these songs to females. So this is mirroring that anatomical data that I had talked about that showed that the duds have fewer neurons here. Basically, we made a dud by going in and pharmacologically knocking down this cell population. So from this study, we can conclude that the BST and vasopressin circuitry does indeed serve to promote male-female affiliation while concomitantly offsetting male aggression. And that's important in this species given that males do tend to be more aggressive than females. So we can see that this circuitry is involved in promoting reproductive social behavior, but I still wanted to know if it was involved in promoting that non-reproductive sociality. So again, I took some male and female zebra finches, and they either got the antisense to knock down the cell group, or they got the control. And this time I tested them in a group size choice paradigm. So this allowed me to get measurements of social contact or general affiliation and gregariousness, which is a preference to affiliate with a large group. So to do this, you place your subject in a cage containing 7 perches that contains a cage of 10 same-sex conspecifics on one side, 2 on the other. The total amount of time spent on those 2 end perches combined yields a measure of social contact or general affiliation. And then the percentage of that social contact time spent specifically with the large group yields a measure of gregariousness. With your normal zebrafinch that's unmanipulated, they usually don't, they're not just hanging out on that end perch, they're usually like clinging up on the side of the cage. They're extremely social, trying to get as close as they can to the other birds, and they're busy singing through the wires at each other. However, when you knock down the BST and vasopressin cell group, we find that in males, but not females, that we see a decrease in gregariousness. So on our y-axis here, we have gregariousness, and this is post-antisense testing minus pre-antisense testing, so that we could account for individual differences in behavior. And we can see that our males that had the antisense and had this cell group knocked down are spending significantly less time next to that large group compared to the scrambled males. So again, earlier in those first studies that we had done about 5, 6, 7 years prior to this study, when we're just looking at the neuroanatomy across species, we see that this circuitry anatomically is mirroring their social phenotype. And now we actually have direct evidence showing that yes, these neurons are indeed promoting that gregarious behavior. What's cool is that when you do this in a different species that is moderately social, you see a really similar effect. And so when we knocked down the BST and vasopressin cell group in Angolan blue waxbills, we actually didn't see any influences on gregariousness, but we did see a reduction in social contact, and again, significantly more strongly in males than in females. So again here, male social contact. And males that had this cell group knocked down were spending more time by themselves in the middle of the cage, as opposed to the control males, which were spending about equal amounts of time next to both the large and the small groups. So these experiments show us that there are similar mechanisms that have evolved to produce a similar social phenotype. And it's not exactly the same. We did see that this cell group modulates aspects of social behavior a bit differently, such that it seems to be directly promoting gregariousness in one species and just general affiliation in the other. However, I still think it's pretty cool that it shows that this cell group is exhibiting some conserved social functions across species. And we can also conclude that those male-specific effects may reflect an evolutionary co-option of circuitry that initially evolved for male-specific reproductive purposes. So in these species, the females don't court the males, it's the males courting the females. And so in those early non-social, non-gregarious species, they still needed to exhibit this courtship behavior, which is why we would see similar functions in the antisocial and highly social species. But then that highly social species, why reinvent the wheel to create a new mechanism to promote a different type of prosocial behavior when you could take advantage of circuitry that already exists? Okay, so these studies tell us about a fundamental mechanism that promotes prosocial behavior in birds. Then I wanted to know, are these mechanisms of sociality conserved across taxa? So enter the prairie vole. Prairie voles are socially monogamous. They're much like those species of birds that we were looking at. They're forming these male-female pair bonds. And they also exhibit biparental care. They are found throughout the Midwest of the US, well up into Canada, which makes them a lot easier to go out into the field and catch them. All those African waxbills, we had to go to Africa to catch them. So having a species that's local to the States makes life a little bit easier. So prairie voles have been famously contrasted with meadow voles. Many of you have likely heard this story before. Our meadow voles, they're promiscuous, such that males mate with a female and then take off. They're also not biparental, so males don't stick around to take care of the offspring. Mothers are the ones that raise the pups. Whereas, our prairie voles, they're socially monogamous and biparental. So there were some really beautiful classic studies conducted by labs of Sue Carter and Larry Young in the 90s showing that nonapeptide receptor anatomy distinguishes these different mating systems. And somewhere along the way, because prairie voles are socially monogamous and form pair bonds, and we like to study them for love, they've acquired this label of being highly social, whereas the promiscuous meadow voles have acquired a label of being territorial. However, just because an animal doesn't form a pair bond, or just because an animal doesn't take care of their offspring, doesn't necessarily mean that they're territorial. And so we have to be careful about how we define social behavior, which is sometimes something that gets lost, perhaps, in the salesmanship that's required to get grants when you're trying to sell your study system. What's great about the vole system for both prairie voles and meadow voles is that there is a really rich natural history studies on their behavioral ecology. So we know a lot about what these different species are doing in the wild. So I dug into that literature, and when we specifically distinguish mating system social behavior from grouping behavior, we see that our female meadow voles are indeed aggressive in the summer, but they're actually quite gregarious and live in groups in the winter. And males are actually gregarious year-round. Whereas our prairie voles are indeed gregarious with their pair bond partners and related kin, but they're actually intensely aggressive with novel individuals. So again, something, I didn't come in here and immediately start brawling with you guys. If I did, not something you would call highly social. So the prairie vole, I would argue, is more moderately social, not highly social. Their sociality is very context dependent. Okay, so flashback to earlier in this talk where we saw that anatomy of this circuit reflects social phenotype. I wanted to know if these species of voles fit that same pattern. And when we do distinguish between grouping behavior and mating system, we do see that they are showing the same pattern. So our more social species, social with novel conspecifics, not in reproductive context, the zebrafinch and the meadow vole, they do both have more of these BST and vasopressin neurons and more receptors in the lateral septum compared to the less social species. So this, again, suggests that, okay, great. So maybe this isn't just a fundamental mechanism for the birds I looked at, but there seems to be some conservation going across taxa. It's also a good lesson to always consider the behavioral ecology of the organism that you're studying. If there is information about what this animal is doing out in the wild, it's always a good thing to have, because what they do in the wild may be very different from what they're even allowed to do in the lab, just based on living situations. And it's also a reminder that we just need to be careful when we define the terms we're using to describe anything, and certainly for behavior and something as broad as social behavior. Okay. So, nonapeptide anatomy seems to be conserved across taxa, but what about function? So are these neurons responding to social stimuli in the voles in a similar way as they do in the birds? So again, we can return to our immediate early gene studies and find out what these neurons are responding to. I'm going to go a bit quicker through this since you have at least seen this sort of study before. So much like we had exposed the birds to a water bath, an empty cage, or a novel conspecific, we can do something similar with the voles. However, voles are much more anxious than a bird. You need to give them time to acclimate to any kind of testing cage. So for an immediate early gene study with voles, they hang out for about a half hour, and then you can expose them to either that empty cage, to something that they really like, which is non-social, which is a wooden block that they love chewing on, or to their same-sex sibling. And so because a prairie vole is not super duper social, and they'll fight a novel conspecific in order to find out if it's, prosocial behavior, we chose a social stimulus that we know is going to be positive to them, which is going to be their familiar cage mate, who is their sibling. And much like we saw in the birds, here we've got that activity of this neuronal cell group. We see that it does indeed light up specifically to the positive social context, and you can't tell a difference in activity between the empty cage and that positive wood block. Okay. Now, with the prairie voles, because they are not so social, I was still curious, what is this cell group going to do in response to an animal that they are going to fight with? So we can see that, yes, it's lighting up in response to positive social stimuli, but is it still promoting prosocial behavior, even in a context that may have more of a negative valence to it? So here, this was part of a developmental study where we looked across age, which I'm not going to show you the ages. Basically, what we did was we just exposed animals to a novel conspecific that was same sex. They certainly did fight. However, there is still some prosocial behavior that they exhibit. And we scored their behavior so that we can relate what is the brain doing in relation to behavior. On our y-axis here, we have the time spent in prosocial contact. So that can involve huddling, positive social investigation like sniffing, or just even kind of positive side-by-side contact. And the more this cell group is activated, the more prosocial the animal. Similarly, on our y-axis here, we have the time spent being aggressive, and the less active this cell group is, the less aggressive the animal is. Yes, am I saying that correctly? Yes. Okay. So all of this together, kind of starting to sound like a broken record up here, suggests that non-reproductive, prosocial, and anti-aggressive functions of the BST and vasopressin cell group are highly conserved. So it certainly can seem a bit redundant to be doing similar studies across species. However, I've been slowly building up on the side a menagerie of animals that are all showing this conserved function. We've seen it in several species of birds, mice, chickens, and now prairie voles. So I personally think that's kind of cool, that this really is suggestive that there may be a central tendency that this circuit is involved in that is going to be fundamental for promoting aspects of prosocial behavior across species, across taxa. Now, for the last few things that I want to leave you with is something new that we're doing in my lab that is still kind of related. So having worked with birds for about a decade, I got really attached to trying to understand what in the brain is promoting that kind of extreme gregariousness and that sociality. And while prairie voles are wonderful for studying things like pair bonding or parental behavior, they're not ideal for studying sociality. So recently, I have brought in African spiny mice to my lab and have established a breeding colony. So I want to tell you a bit about these unusual little mice, other than they're really cute and have unusually gigantic eyes and ears. So spiny mice, they are found throughout Africa, the Middle East, as well as Southern Asia. And what's kind of cool about them is most rodents that you think of, they burrow. And so spiny mice do not dig burrows, and they don't live underground. So they like living in rocky outcroppings and crevices. This is a good thing for me because we can actually go out into the field and learn something about their behavior. You hear about people going birding all the time. I like birding. Go out there with your binoculars, beautiful birds flying around. They communicate the way we do with primarily auditory and visual methods of communication. So birds are pretty cool because we can look at them and have a good idea of what they're doing. You can tell when a bird is really angry. Rodents, people don't go rodenting because rodents are typically underground. There's not much to see. They're wandering around in a field looking for holes in the ground. And so what's great about this species is that because they're living above ground, there's a lot more information that we can get about their behavioral ecology with relative ease. So spiny mice are primarily studied because they are the only mammal known to date that can completely regenerate entire suites of tissue. So this is pretty crazy. They can regrow hair follicles, skin, sweat glands, fur, cartilage. The tensile strength of their skin is 21 times weaker than that of your average mouse. So up here in this picture, this is what happens if you try to scruff a spiny mouse. So if anybody's ever worked with rodents before, usually to pick them up, you're going to kind of trap them under your hand and scruff them, much like you would scruff a cat by the nape of its neck. And if you do that to a spiny mouse, you will rip all of their skin off. And then they must have crazy immune systems, of which nobody is yet studying, because they can run around like that in the wild for 2 to 3 weeks, and then they grow all of their skin back. And it's not scar tissue that's growing there, it's the skin regenerating. So pretty cool. Totally not my area of study, but that's what people study the spiny mice for. Yeah, ridiculous. Okay, so cool mice. Oops, and also I apologize, I've got different species names up here because last week we had the genome resequenced and realized that we have a different species than we thought we did. So this is the kind of fun roller coaster of when you work with a relatively novel animal to research, is that there are only 12 labs in the world that work with these guys. We're the first to do anything social behavior and certainly social neuroscience, anything neuroscience. And just got the email from people that I've never met before in Israel saying, like, actually, you don't have Acomus caerurinus. You actually have Acomus demidiotis. So good thing we haven't published yet. That aside. All right, so spiny mice. I'm not a regenerative biologist. Why do I care about them? So the spiny mice are unusually highly social. So from those biologists that have gone out to trap them in the wild to start their breeding colonies, they've reported that they're living in groups of 20 to 50, comprised of related and unrelated individuals. In the lab, we can house them in cages of 20 to 30. And so, it's a cage about this size, and they're super cooperative. You take a random novel male, throw it in the cage, and they'll just kind of like, come on down, welcome to the huddle. That doesn't happen with a lot of rodents. So these guys are really highly social, which is right up my alley. They're also cooperative breeders. So any adult or young adult that's walking around and sees a pup, they'll just take care of it. They're also the first rodent that is known to menstruate. So they're bizarre in more than one way. And then also what is cool for my interest is that they're precocial. And so they're born with their eyes open, ears open, smell. They can independently locomote as soon as they're born. They can eat solid food on the first day of life. And so if you're interested in, okay, what are the early life experiences that are shaping these adult social phenotypes, one could imagine that there might be some different processes going on in an animal that can start engaging in social interactions right from the get-go, versus most animals are altricial, and they are completely helpless little shark bait just attached to their mother for like the first 2 weeks of life. So there's some cool neuroplasticity questions that we can ask in relation to the development of the social brain. So because of that high degree of sociality, spiny mice are a much more appropriate organism for me to be using to look at what it is that makes large groups function well, and what it is about the group phenotype as well as the individual phenotype that can result in that kind of peaceful coexistence in a large group. So a little preliminary data for my beloved BST and vasopressin cell group. It's kind of sounding like an unhealthy obsession. This is not a y-axis, but it kind of looks like it. So over here we've got the average number of vasopressin neurons in the BSTM, and then the different species that I've been looking at. And we can see that our kind of less social species, the more social we get, the more neurons we have. I was quite delighted to see a nice robust population in the spiny mice. And for comparison, the difference between that really territorial bird and the highly social bird. So suggesting that, okay, woohoo, maybe there is that conservation keeps going, that central tendency really might exist in reality and not just in my head. So that's exciting. Other early studies with the spiny mice. So no one has yet to systematically characterize their social behavior. So that is what we are currently doing. And then also using immediate early gene studies to find out what these neurons respond to. And then we're also using viral vectors because antisense technology is outdated. And so we've got some nice AAVs and dreads that are working in them, which is great, so that we can go in and we can manipulate those circuits. And then what's really neat is because of this dread technology, for the first time, instead of just looking at the BST and vasopressin and the lateral septum independently, we can actually manipulate those direct projections at the same time. So to wrap it up, I talked about really just one little subcircuit in the brain that does show functional conservation across taxa, but that's also just one circuit in the brain. I'm definitely not saying that this is the only thing in the brain that promotes prosocial behavior. Absolutely not. Brains are incredibly complex, just as social behavior is incredibly complex. And so this is just but one tiny piece of the story, and why I also love coming to give talks like this and to meet different scientists to find out the different sorts of approaches that they're taking and the different parts of the brain that they're looking at to understand what makes animals do what they do. With that, acknowledgements, all the bird work was conducted at Indiana University, the vole work, the majority of it at Cornell, some of it here at Emory, and now the spiny mice at Emory. Also a shout out to my collaborator, Ashley Seifert at University of Kentucky, who's the regenerative biologist that I would not have these spiny mice if it weren't for his generosity and openness to help me start my own colony. With that, I can take any questions. Yeah.
You showed a difference between the studs and the duds. What made the studs successful? Was it a qualitatively different behavior, or just did they try more?
It's mostly so the number of dances that they do and the number of songs, and then we also did look at what's the outcome of that is the female receptivity. And so when males start doing their little dance and their song, the females will solicit copulation, where they kind of like, it's kind of like lordosis in a rodent, and so they'll like lift their tails and lean forward. And so that would be certainly considered as a success of courtship. Yeah.
I was wondering about timescale of how variable the behavior is, and if that's conserved.
Of the social behavior or like sociality?
If you put in the antisense and if you turn a stud to a dud, you promote one behavior and suppress the other behavior,
Yes.
is the timescale radically different, or is the timescale also potentially sort of conserved?
It's all, so with the antisense, it's kind of all overlapping. So it's almost like you're altering the complete phenotype. So whereas something like dreads and opto, you're on the timescale of like milliseconds with opto to minutes and hours with dreads. With the antisense, it's more kind of like a short hairpin RNA, where you're just getting a constant, consistent knockdown of behavior. And so you're just making basically, now for as long as you keep infusing this antisense every 12 hours, you will have an angry bird just all the time until you stop infusing the antisense, and then production starts to ramp up, and then their behavior changes again. Does that answer your question a little bit?
So I guess the earliest time points you would have would be these like 12-hour time points when everything is sort of steady state knockdown versus looking at faster timescales.
Yes, right. And so with them, before we did any of the behavioral tests with the antisense, first we did the timing of after the first infusion, how many infusions do you need? How many 12-hour cycles do you need until you see a ceiling effect of what it's going to do? And so that knockdown is about 55%, and it's after 72 hours that it just seems to stabilize. And so that's when we would start testing, so that at least we kind of had a consistency in how their phenotype has changed. But I'm super curious with things like dreads to be able to see how quickly you can kind of go back and forth with the behaviors. Certainly there are some amazing opto experiments where all of a sudden they turn on the laser and super aggressive, and so I'm not certain if we'll see similarities with that here. Sorry.
Has there been any research on monkeys or humans related to these regions? Obviously not kind of invasive work, but anything else?
Surprisingly, not to my knowledge, really. So most people that are looking at the BSTM are not specifically looking at vasopressin in primates, and they're just looking at it generally in relation to anxiety. So most people in rodents have found that vasopressin is anxiogenic. And in the birds, we've actually shown that it's anxiolytic. And so that might just be a general difference between taxa. But again, not so much in humans and primates, because people don't look at vasopressin there. It's a wee random region of the brain, but it's connected to the amygdala, which of course everybody's interested in.
So you mentioned you looked at several different species. Can you sort of talk about what are maybe the most extreme species, and what would be like some dream species that might be out there that maybe would show, oh, this is really generalized across the kingdom, or?
Yeah, I mean, it would be amazing to look in a highly social primate, which is a non-human primate, would be feasible depending on the right collaborations. Looking at so it took me a while to search for a mammal that was highly social that I could bring into the lab. So it's like when you think of highly social mammals, I think of ungulates, like wildebeests, like they're in flocks of, herds, not flocks. Wildebeests are in gigantic herds running throughout Africa. And so even, a lot of caribou and reindeer, like all these giant ungulates you see on the Discovery Channel doing their really cool collective behavior, those coordinated movements. And so that type of sociality, to be able to even move in coordination like that, is different. That could be a really interesting system to look at to see if sociality and kind of large-scale coordination like that are modulated similarly. One could do that to a certain extent in fish, but they have slightly different anatomical morphologies such that they don't really have a BSTM containing. For them, it would be vasopressin cells. So you'd have to look at different nonapeptide populations in fish.
Like dolphins.
I don't know about dolphins. It's a good question.
Here's a question from online.
Oh, from online. Oh.
Were the number of VP neurons in the BST and absolute numbers of neurons? Elaborating on the methods would be helpful.
Yes. So at first I was digging through the literature to try and find papers that were reporting cell numbers in a manner that was consistently collected, such that a lot of people will take every 120 microns, they're going to take a snapshot of the brain and count those. And it started to get a bit too unreliable for my taste. And so I decided to just get as many brains as I could and do it myself. That way it was done the exact same way by the same person with the same antibodies. And then, yes, they are absolute cell numbers. So it's not that large of a cell group. In the birds, it's pretty big. Those zebra finches, I mean, there are tons of them. But even in the prairie voles, like 30 neurons is really not that many. And especially in animals where they have fewer and fewer of these neurons, that's a small target to go in and hit with something like an AAV. But yeah, so absolute numbers in the BSTM.
First of all, I totally think you should bring a herd of wildebeest in.
I'm going to try. If you see me on the news.
I really like the comparative approach, and I was curious from the evolution perspective then of this within a species, how much of the variation that you see intraspecifically is heritable versus plastic? Is anyone? I've heard a few other studies that have looked at like comparative magnitudes of expression of this, and I haven't ever heard anyone talk about that.
Yeah.
I'm sure it's out there.
For this cell group in particular, I don't think anybody's really looked at it. I have looked at individual variation in septal nonapeptide receptors. It was in the voles, and it was a epigenetic study where prairie voles were raised with single mothers or two parents. And then also some of the mothers were working, so we wanted to see how, try and mimic working parents. So basically we made the parents work for their food. So anyway, but you do see that based on that early rearing environment, you can see big differences, mostly in males. So with females, they had pretty stable anatomical outputs, at least. And you would even see methylation differences in both females and males, but you only saw the change in gene expression in the males. And in a couple other studies that we've done, there seems to be something particularly plastic or susceptible about males and not in females. I'm not entirely sure why that's the case. But that's about as much as kind of we and others have done on the plasticity end of things.
So do you think the circuits are actually evolving, or just the plasticity of the circuits?
That's a good question. I think over really long periods of time, when you see something that suggests that co-option, that the circuits themselves might be evolving, but maybe not. I mean, it could be reading too much into it. It just could be that in those highly social species, the circuits simply need to become more plastic so that they can be used for more things.
Okay.
Thank you.
Reminder that we do not have seminars until 20 October, so we have a break for a couple weeks. So prepare your lunches for your Mondays.